All right, in Chapter 7, we're going to look at the control of microbial growth. In Chapter 6, we talked about the requirements of microbial growth. Now we're going to look at how we can control it. So some definitions that we need to know are bactericidal versus bacteriostatic. Anytime you add "cide" or "cidal" to the end, this is a suffix that indicates destruction of a type of microbe. Okay, so when we say bactericide, it's going to kill bacteria, fungicide kills fungus, and so on. This does not necessarily take care of the endospores, though. On the other hand, when you add stasis or static to the end, this is a suffix that indicates inhibition, but not complete destruction of a type of microbe. So it prevents it from replicating and growing, but it does not kill them. All right, this would be bacteriostatic, meaning that it's going to inhibit bacterial growth. Fungal static is going to inhibit fungal growth. Virostatic are inhibiting viruses. All right, and so when we look at this, there is a difference between it being cidal versus static. All right, some other things we want to look at are some terms like sterilization. Sterilization is the destruction or removal of all forms of microbial life. Now, this does include endospores, okay? But with the possible exception of purons or prions. Because of this, they're harder to potentially get. Sterilization may not take care of them, but it does take care of endospores. If we look at disinfection, this is going to be the destruction of vegetative pathogens. It's not going to take care of endospores, but it will take care of the vegetative pathogens. We also have antisepsis. This is destruction of vegetative pathogens, but this is going to be used on living tissue. All right, so there is a difference between a disinfectant versus an antiseptic. So an antiseptic, guys, is mild enough to be able to be used on your skin. Some examples are hydrogen peroxide, mercurochrome, tincture of iodine, and 70% isopropanol alcohol. On the other hand, a disinfectant is going to be used on inanimate objects. Examples of this are going to be bleach, heat. We'll also look a little bit at commercial sterilization as well as degerming and sanitation, which are all just a little bit different. Some of these types of microbial growth control are better than others. There are some factors that influence the effectiveness of an antimicrobial, something that works against microbes. First, we have to know the number of microbes. The greater number of microbes, we have to increase the killing time. And so because of that, you'll have to increase the amount of time that chemical or that heat or whatever it is that you're using is in contact with those microbes. Another thing are some environmental influences, the idea that it's in body fluids. If it's found in body fluids, you also need to increase the killing time. Time of the exposure, the longer time that you expose it to, the increase of its effectiveness. So if 10 minutes is good, 15 minutes might be better. Now, of course, there is a fine line where it's too much when we're talking about hurting the potential object that it's on, okay, or in. But when we're looking at this, though, increased time normally increases its effectiveness. Another thing is the characteristics of the microbe itself. And so when we look at these, microbes are kind of in a hierarchy. Some are more susceptible to antimicrobials than others. These include things like viruses with lipid envelopes, gram-positive bacteria, viruses without envelopes. These are all very susceptible to lots of types of antimicrobials, but others are more resistant, like endospores are more resistant as well as the purons. So there are some different actions that a microbial control agent can undergo. It's either going to affect protein synthesis, it's going to affect the DNA or the plasma membrane. So one thing is it could alter the membrane permeability. This could damage the membrane lipids or proteins, which diminishes the cell growth because it damages that ability of being able to regulate what moves in and out of the cell. Another thing we can damage is the proteins. It can denature the proteins by using heats or chemicals. And remember, when we denature a protein, it unfolds and it's no longer usable. Another thing we can do is actually damage the DNA and or RNA that's present in the cell. This is where you're damaging the nucleic acids. This hinders replication so that it can't make more of itself, and it also will slow down protein synthesis. So even though the proteins are working properly, if there's no DNA and RNA, then it's going to slow down that process as well. Some of these microbial control factors for growth are going to deal with the physical side, and some of them may be chemical methods. All right, what type of equipment or surface are you dealing with? This is going to help us decide whether we need to use something that's physical or chemical. Is it heat liable? Can you actually heat it? Some stuff cannot be heated up, like plastics. You can't heat that up too high, otherwise they'll start to melt. Is it disposable? Again, is it a type of plastic? Is it a metal? What type of microbe may need to be killed? All right, is it do you think gram-positives are present? Viruses, purons, what do you think might be present there? Sterilization of media and pharmaceuticals are important. However, do they have a high protein base to them? Do those proteins important for them to be active? If it is, you cannot use a high heats because it denatures those proteins. And so again, you have to look at a lot of different factors to determine what kind of method you're going to use. There are established recommendations or protocols that are kind of put together in order for you to follow. Some examples of some physical methods include heat, filtration, even using cold, desiccation, osmotic pressure, and radiation. So we're going to take a look at some of these. All right, so if we're looking at heat first, these are again physical methods. When we're looking at heat, we can look at boiling. We can also look at autoclaving. We can look at pasteurization, and then also dry heat. Now, when we're looking at boiling or autoclaving, you'll notice that this does protein denaturation. So we see here that it's going to denature the proteins and make them where they cannot be utilized. Then we see here another thing that's important on this chart is the comment of that kills endospores. So if you look at boiling, it kills vegetative, bacterial, and fungal pathogens and almost all viruses within 10 minutes, but it's less effective on endospores. All right, so boiling might be good for some things, but not others. Autoclaving is a very effective method of sterilization at about 15 psi of pressure. This can bring the temperature up to 121 degrees Celsius. This will kill all vegetative cells and their endospores are killed in about 15 minutes. So if you're going to use one of these methods, autoclaving might be the better method versus the boiling. Pasteurization is a heat treatment for things like milk. It brings it 72 degrees Celsius for about 15 seconds and it kills all pathogens and most non-pathogens, those that are responsible for spoilage. Okay, but if you'll notice, it does not talk about endospores. We also see in this chart, it shows you preferred uses. When would we use these types of heat? Another thing deals with the dry heat. When we're looking at dry heat, you can do it directly over a flame. This would be like Bunsen burners or things like that, but normally it turns the contents to ashes. Also, incineration also burns things to ashes. So if you're wanting to keep certain things, then we obviously don't want to use that. Another way is like with hot air sterilization, which is like an oven. This is very effective method of sterilization, but it requires temperatures of 170 degrees, but it's for a long time, okay, for 2 hours. Okay, and so we're looking at this, it could take a lot longer. All right, so the big thing is looking at what they can do, moist heat versus dry heat. All right, so what we see in lab is a lot of times we're going to be using autoclaving or direct flaming. The direct flaming is what you will have seen in lab where they will take the inoculating loop and things like that and put it directly into the flame. Or you'll take the slide when you want to heat fix it and put it directly towards the flame. On the other hand, autoclaving is how we get rid of a lot of our waste, making sure again that everything is killed before we throw it in the trash. In lab protocol, if you were in a face-to-face lab, if anything gets contaminated with the bacteria in lab, it has to be autoclaved, and this includes your clothes, your book, things like that. So it's very important that you are careful, and there's no guarantee it's going to come out the way it went in, especially if we're talking about a book, because we're using high pressure and also water in order to get that high temperature. So when we look at heat, heat denatures proteins and kills endospores. Moist heat requires shorter time as compared to dry heat because water conducts heat better than the air does. All right, and so since the water can conduct that heat, it's going to have less time to be used. When we look at pasteurization, there's classic types of pasteurization. However, there's some more advanced types now that don't take as long. One is called the HTST FT or flash type of pasteurization. This is known as high temperature short time pasteurization. This decreases the bacteria, but it does so if you will immediately refrigerate afterwards. The other one is UHT, and this is ultra high temperature, or also known as ultra pasteurization. All right, and so this is using really high temperatures. The higher temperature, the less time is needed. Okay, and so if we can increase the temperature, we don't have to put it in there for as long. This does not achieve sterility when we're looking at pasteurization, and the whole purpose of pasteurization is to kill pathogens and to decrease the microbes associated with spoilage. It doesn't say it will get rid of them, but it does decrease them. That way the shelf life of it can be longer, like with milk. One of the main ways that we use heat in the lab is through a steam autoclave. The whole point of the steam autoclave is it allows you to use really high pressure and hotter temperatures to sustain the high heat. This is better than with boiling because it actually will keep it in a contained area. This is effective if material is to be sterilized if it's not damaged due to high heat or moisture. All right, so like with glass, this one is an easy way to use that sterilization. The glass still comes out looking the way that it should, and so when we want to sterilize water or pipettes or things like that, this is used in laboratories and healthcare institutions to sterilize culture media, solutions, and instruments. And so they'll use a steam autoclave like in a surgical ward in order to kill the bacteria or any microbes that are on it, and then they repackage it and seal it, and then it has been sterilized. We also remember, if you remember from the chapter about the media, it's always important to sterilize it before you inoculate it to make sure nothing else is growing on it. So in the lab, if you're wanting to get rid of the biohazard waste, it's really important in order to do quality control to make sure that the stuff has died or is dead. One way you can do this is through strips. This one has a type of organism that is known embedded in the strip, and if it is killed, then you see that it gives you a color type of indication. Again, quality control is really important, especially before you throw something in the normal garbage, because you don't want it to potentially infect others. So when you're looking at steam autoclaves, they work on moist heat, pressure, and time. And so this particular diagram shows you kind of how it works. When we see this, if you're using a certain amount of pressure, it will raise it to a certain temperature, and then the time can be adjusted. And normally the standard is about 15 psi, which brings it to about 121 degrees Celsius, and this needs to run for at least 15 minutes. Now with dry heat, we use dry heat in the lab when we're flaming loops or forceps. We do this in order to sterilize them so that they can be used. We also see that you can use incinerators. These incinerators can sterilize. It destroys the material that is there. And so if you've ever used one of these little incinerators in a lab, if there's bacteria on your loop, you can actually hear it popping, okay, because it's killing it and then it gets removed. Again, hot air ovens could potentially be used, but the time has to be extended due to it being a dry heat. Another thing we can look at is filtration. Filtration, we must sterilize the equipment prior to doing the filtration. So all the equipment needs to make sure it's been sterilized. You can maybe use an autoclave, an oven, things like that. Passage of the liquid through membrane with pores too small for microbes to pass. All right, so the liquid can pass through, but the microbes cannot. We use a vacuum, which helps force the liquid to pass through the filter into the flask. It's used on heat-sensitive liquids. And so these are liquids that you can't autoclave or you can't use in like an oven or boil. So this is really important in separating bacteria from its suspending liquid. A note here is purons in a mycoplasma may be filterable. They are very small in size, and so we could potentially see that they are filterable. This is useful for sterilizing liquids, again, those especially who have enzymes or vaccines, to make sure that these things don't get destroyed by heat. So we have methods that deal with cold, things like refrigeration. We also have what we call desiccation, and we also have osmotic pressure on this particular chart. So this shows you the preferred uses. When we're talking about the cold side of it, we can use this for food, we can use it for drugs, and even culture preservation. On the other hand, desiccation and osmotic pressure is more for the food side, okay, in order to preserve food. One thing to note about refrigeration is that it is bacteriostatic. It is not bactericidal, but it does have a bacteriostatic effect. Desiccation and osmotic pressure, these guys are going to help disrupt either metabolism, like in desiccation, or osmotic pressure is going to cause plasmolysis. So let's look at the low temperature first. So with the low temperature, the effect varies per microbe. Some microbes, recall from previous chapters, they actually grow better in the cold temperature. Therefore, this would not be an effective way to control them. This is only bacteriostatic. It inhibits their metabolism. Refrigeration also reduces spoilage, but freezing may not kill all the organisms, but it could kill some of them. The idea with the freezing is that it can cause them to freeze up. That doesn't actually kill them, but when they start to thaw out, the water changes due to that thawing could potentially hinder the growth or even hurt the bacteria. The next one is the desiccation. Desiccation is the absence of water. This is going to inhibit metabolism. Microbes can remain viable and dormant for many years in this. The effects again can vary based on the microbe. So some might be affected more by desiccation. They dry out and they no longer can affect you. Others can last longer due to like an encapsulation or even the endospore that's present. So when we look at these examples, the Neisseria gonorrhea, this one can last for about 1 hour after it starts to dry out, whereas the tuberculosis can last for months and endospores can last for years. We also then see osmotic pressure changes. This is achieved by hypertonic environments. This could be with high salts and sugars. This causes plasmolysis. This is used a lot of times when we talk about canning or even preserving foods. Like with salts, it would be more for like meats. When we look at sugars, that's more for your fruits. This is one reason why you'll see like if they're put in the little jars and stuff like that, they contain the high sugar, and it's to help decrease the growth of microorganisms. And the point here is to inhibit their metabolism as well. The next main method here is radiation. When we're looking at radiation, we can use ionizing radiation or non-ionizing radiation, but it doesn't matter which one you're looking at. Both of them do affect the DNA. Ionizing radiation, though, destroys the DNA, whereas non-ionizing just damages it. Okay, and so this is what we're looking at. Now, ionizing is not widespread in routine sterilization, but they do use it to sterilize some pharmaceuticals, medical, and dental supplies. The non-ionizing is the radiation is not very penetrating. That's one reason it's not used, but this is to control closed environments with like UV lamps, and this is more germicidal, all right, and so it's to kill the germs that are present. So with radiation, it can kill microbes depending on the wavelength, the intensity, and the duration that it's exposed to this radiation. When we look at ionizing radiation, this is going to be the radiation that has the higher intensity, the shorter wavelength. These are going to include the gamma rays and the X-rays. They can be used to sterilize heat-liable drugs and medical equipment, but the reason for this is they do alter the DNA. This is why you should not expose yourself to gamma rays and X-rays. If you get X-rays occasionally, it's not that big a deal, but that person who is the X-ray technician, they need to protect themselves if they're giving hundreds of X-rays a day. All right, and so these are the ionizing. The non-ionizing would be more like the UV lamps. They're there to damage the DNA. They produce a pyridamine dimer, where we see that if there is a thymine next to a thymine in the DNA strand, it actually causes them to pair with each other instead of pairing with A like they're supposed to. The UV light is good for surface decontamination, and so this is what we see, and this is one reason why UV radiation does potentially cause skin cancer for us too, because it does damage our DNA, and in the case here we would use it to damage the microbe's DNA. Another type of the non-ionizing radiation are microwaves. Microwaves have a very low energy wave. It does not have a direct effect on the microbes, but the high temperature from the water does. When we microwave food and things like that, we're actually heating up the water molecules that are found in those substances. This is what causes that heat to happen, and that's one reason why it's really uneven when you have that heating and you have to kind of stir it up. This will also start to affect the microbes too, based on how much water is present. All right, now switching gears, we looked at the physical methods. Now let's look at the chemical methods. Chemicals can reduce bacterial populations or remove vegetative forms. A lot of times they're non-endospore forms. They're not going to affect the endosperms. These generally don't achieve sterility. Unlike what we saw with the autoclave and things like that, we're not going to actually sterilize this. There are different disinfectants that are effective for different microbial groups. We find that some things like bleach will work better for some microorganisms, but not others. All right, so the first group of chemical agents we want to look at are the phenols. Okay, when we look at this, you have the phenols, the phenolics, and the bisphenols. When we look here, the phenols, all of these are going to look at disruption of the plasma membrane. They also are going to help with denaturation of enzymes. Now, guys, phenols are rarely used except as a standard of comparison. These are seldom used as a disinfectant or antiseptic because of its irritating quality, like on the skin, and it has a really bad odor to it. The phenolics are going to be for environmental surfaces like instruments, some skin surfaces, and mucous membranes. This is a derivative of phenol that are active even in the presence of organic materials. Bisphenols are going to disinfect. They're going to be disinfectant hand soaps and skin lotions. Triclosan is especially common example of this. It's a broad spectrum and it's most effective against gram-positive bacteria. So guys, when we look at these groups, phenol was actually used by Lister. And if you remember when we talked about him in Unit 1, he was the first one that started disinfecting. And he was the one that was trying to reduce the idea of the childbirth fevers and also during surgeries and passing on infections. However, he found that this is very irritating to the skin and it did have a really bad odor. This then led again to the phenolics. This is not as irritating to the skin. One of the main groups is what we call cresol. It's the main ingredient in the disinfectants like Lysol. And then the bisphenols are like the triclosan. This is in antibacterial soaps. We also have the hexachlorophene. This is found in some prescription or surgical scrubs. And this is especially effective again against the gram-positive bacteria. We see triclosan, if you look in these pictures, can also be used in toothpastes. All right, so if you look here in the toothpaste, you can see Colgate has that, whereas Crest does not. One issue with using the triclosan too much is if it can cause widespread, if it's used in a widespread way, it can generate resistance of bacteria. So we have to kind of be careful on that use as well. Now, in order to determine if these are present, you have to look at the label. And this is, again, why labels are located here on this slide. So the next group are the biguanides, also known as the chlorhexidines. These are going to disrupt the plasma membrane. We can use these for skin disinfection, especially for surgical scrubs. These are bactericidal to gram-positives and gram-negatives. These are non-toxic. And they are persistent. And so when we look here, some examples, this is showing you the antiseptic type of mouthwash, and we also have some other cleaners. And again, you would be looking for that chlorhexidine on the label. The next group are the halogens. The halogens are effective against all kinds of bacteria. They also are effective against their endospores, viruses, and fungi. The main thing that these guys do is they're going to help inhibit the protein function. There are two main forms here. The iodine is going to be used as an antiseptic, and the chlorine is going to be used as a disinfectant. So recall earlier in this lecture, an antiseptic means it's going to be used on living tissue, and a disinfectant is going to be used on inanimate objects. All right, so this is what we're looking at. Now, this first particular slide, it's showing you the iodine products. All right, so the first one here is the povidone. The povidone is a lot of times used in first aid, and the whole point is to decrease wound infection. We also see betadine. Betadine is also used in wound disinfection. It also can be, when we look at iodine especially, these could be used to treat water. Campers sometimes use this to help treat the water before they drink it. The last one here is the tincture. This is do not stain, and it's less irritating to the skin than the other types of iodine. It's put into an aqueous type of alcohol solution, and it is released more slowly. Chlorine products are a little different. We have the bleach. The bleach has sodium hypochlorite in it. We also see the chloramines. This is chlorine in combination with ammonia. One thing with this group too is it doesn't leave a residue, and it's odorless and tasteless. The next group are the alcohols. These include things like ethanol and isopropanol. These are going to do protein denaturation and also lipid dissolution, meaning that it's going to dissolve the lipid components, the fats. We use these a lot of times with thermometers and other instruments. We do this also to swab the skin with alcohol before we give a shot or something like that. And the whole point there, though, is it's simply wiping it away. And so it's more of a degerming than it is an actual like sterilization. This is bactericidal and fungicidal, but not effective against endospores or enveloped viruses. And so again, we can use this. This is rubbing alcohol. This is less volatile than just normal alcohol, and this is why they have a different percentage to them. Now, these percentages are kind of important because we find that if the percentage is too low, like 40 or even 50%, we see that the growth still happens with a lot of these types of bacteria. No growth starts to happen at 50% if it's been left on for 30 seconds or longer. If you look here also at the 100%, if you use 100% ethanol, there's growth with the time of exposure. And so 70% has been found to be the most effective percentage for denaturing the proteins. And so this is why most of these products on the market are normally a 70% ethanol concentration. And the reason this percentage is less than 100% is that in order for denaturation to take place in the proteins, water has to be present. And if it's 100% ethanol, there's no water that's present. All right, the next group we're looking at are the heavy metals. Heavy metals can be used in very small amounts, and they can kill or inhibit growth. The way they do this is they denature enzymes and other essential proteins. Heavy metals such as silver and mercury are biocidal. So when we look at this, some examples are things like silver nitrate, muric chloride, and copper sulfate. And so these heavy metals help in, again, inhibiting or killing the bacteria. Some heavy metals have antimicrobial effects. So if you look here in this example where they put coins that had different metal concentrations on the bacteria or on the spread plate, you can see that some of them killed or at least inhibited the growth, whereas others didn't. Inoculating loops are made of platinum, tungsten, or nichrome. So this way it does not kill the bacteria, because if you use something that had silver, copper, or something like that, it would kill the bacteria before you could transfer it over. Now, when we look at this, guys, the mercuric chloride is very toxic. It's corrosive. And so it's ineffective on organic materials. The chloride, though, also tells you that it has a kind of bacterial static type of structure or a role. And so this would be used more as a disinfectant. Thimerosal is an example of an organomercury compound. However, this is, and you can see it here on this label. However, newer products on the market are actually mercury free, even though they kind of show that they seem to have mercury in them. They're actually mercury free, and you can look at this on the label because there's no mercury contents, and they actually contain a different type of chemical that's called a quat, which we'll talk about in a minute. On the other hand, there are many different things that use silver that are on the market. One is the silver self-design. This one is going, this is going to be used a lot of times on burns. We also see that you can use silver nitrate solutions. These are used as a prophylaxis on babies. If you'll notice, once babies are born, they put these eye drops in their eyes, and the whole point is to help be proactive in making sure that there's no infections of the eyes. Especially when they go through that birth canal, there's a chance that they can be exposed to lots of different types of bacteria or viruses, and so we want to potentially protect the eyes. Now, silver has actually got an increase in its interest because we do find that it can work against antibiotic resistant bacteria, so potentially coming up with types of medications that have silver in them that can be used against those instead of antibiotics might be helpful. Now we want to look at the surface actives. They're also known as surfactants. These are things like your soaps and detergents, and also the quats. The quats are what we call quaternary ammonium compounds. When we look at soaps and detergents, this is more of a mechanical removal of the microbes. This is more through scrubbing. It causes skin degerming and removal of debris. The quats are going to do enzyme inhibition, protein denaturation, and disruption of plasma membranes. It could be used as an antiseptic for skin, instruments, utensils, or even rubber goods that cannot necessarily be heated and things like that. So let's look at soaps and detergents first. These are what we consider emulsifiers. They do not kill or inhibit the growth of the microbes, but they mechanically mechanically remove the germs from your hands or from the surface through scrubbing. It breaks the film of oil, dead skin cells, sweat, and microbes into droplets, and this is what's called emulsification. And then we can rinse them away with water. When you rinse them with water, it lifts and carries away those droplets, thus de-germing the skin's surface. The next group are the quats. These are the quaternary ammonium compounds. This is the most widely used type of surfactants. They have positive charged molecules, which gives the quats their cleansing ability. They actually are really good against killing gram-positive bacteria, fungi, amoebas, and enveloped viruses. However, they are not good against endospores or mycobacteria. They do not take care of those. Again, looking at these labels, you can find them as the active ingredients. When you look at soap or a mouthwash, when you put it in your mouth and you start swishing it around, it causes that foaming action. That is due to the quat that's listed here. This is also known as Isopropanol. We then see that we can see some others here in these example of these antiseptic towelettes. This is also known as Zephyrin. This can help again with cleaning off surfaces. The good thing about quats is they are colorless, odorless, tasteless. They are very stable, and they're easily diluted in order to be used in cleaning supplies, as well as being non-toxic. The next group are the aldehydes. The aldehydes include formalin and glutaraldehyde. These are going to do protein destruction. Aldehydes are actually some of the most effective antimicrobials. One example is formaldehyde. It's usually in the form of formalin. It can be used to preserve biological specimens. So if you've ever taken a biology lab where they've got the specimen in a jar with fluid, it normally has some sort of formalin type of fluid on it to help preserve it. Because if bacteria gets on the specimen, it starts to decompose it. We also see the glutaraldehyde. This is less irritating than formaldehyde, and actually is more effective. It is a bacterial-side, a tuberculosicide, meaning that it can kill tuberculosis, which is the mycobacterium, and it's also a viral-side within 10 minutes. It's also sporicidal in 3 to 10 hours. All right, so if you leave it on longer, it actually can kill the spores as well. Commonly in the form of a 2% aqueous solution, also known as Sidex. It's used to disinfect hospital equipment. And so if you look here, you can see that the Sidex is here. We also see that formalin and our aldehydes are found in embalming fluids. Okay. One thing to notice, formaldehyde is poisonous. It's very dangerous if it is consumed. We then have chemical sterilization. Chemical sterilization is using a toxic gas called ethylene oxide. This inhibits vital cellular functions. Ethylene oxide is a gaseous chemical sterilant, meaning that it's in a gas form and it kills all microbes and endospores. This has to be applied in a closed chamber, and it has to be applied over several hours. It's usually mixed with a non-flammable gas like CO2, and it is also used in hospitals. One thing that they use here is that this gas is very toxic, but it's also explosive. And so this is again why they put it into a non-flammable gas substance with carbon dioxide, because if they mix it with oxygen, it could cause major issues. Chlorine dioxide is used to fumigate enclosed areas that have contained anthrax endospores. So anytime we see that anthrax has gotten access into an area, they would use this to fumigate. This is going to be used a lot of times on larger things like hospital mattresses and stuff like that. The last group we're going to look at are the peroxygens. This is like hydrogen peroxide. Their mechanism of action is through oxidation. Hydrogen peroxide is a sporicidal at high concentrations. It's broken down to water and oxygen by an enzyme called catalase in aerobic cells. We also see that there is parasitic acid, or PAA. This is one of the most effective liquid chemical sporicides. It kills endospores and viruses within or in 30 minutes, and it leaves no toxic residue. It can be used for washing fruits and vegetables. We also see benzoyl peroxide is in a lot of over-the-counter acne medications to again decrease microbial growth, since that's what potentially causes most of the acne. So again, if we take a look here, we have the difference. The antiseptics can be used on the skin. You have the hydrogen peroxide, the Oragel, the benzoyl peroxide gel for acne. On the other hand, we see that they also can be found in some disinfectants like OxyClean, and also Clorox with hydrogen peroxide. And of course, they're disinfectants if they're going to be used to clean inanimate objects. Now, how do we evaluate the effectiveness of these chemicals? How do we know that they're really good against gram-positive or enveloped viruses or different things like that? One thing we can do is we can do an evaluation of a disinfectant using a disc diffusion method. Okay, we do this with antibiotics as antimicrobials as well in chapter 20, which we'll look at later. The paper disc gets soaked in whatever chemical you're wanting to look at, right? It's then placed on an agar plate that's already been inoculated with the test organism. So let's just say we want to test out these chemicals on E. coli, or we want to test out these chemicals on staph. We're going to take the plate and we are going to do a spread plate. We're going to cover the whole plate with the bacteria. Then we're going to take this disc. The disc is going to be soaked in whatever it is. In this case, they did chlorine, they did a quat, they did some other chemicals here. So they put it in there. Then they placed it on the plate. As the plate was incubated, we see that bacterial growth did occur. However, in some of these, it created what we call a zone of inhibition. This is a clear area where the chemical inhibited the growth, when we can't know if it killed it. All right, so in this case, we'd say it's at least bacterial static. We'd have to do further tests in order to know if it actually killed the bacteria and it was bactericidal. All right, so in this particular example, if we look at Staphylococcus, all 4 types of disinfectants or chemicals actually did create a zone of inhibition. However, chlorine's was the largest. For E. coli, though, we see that chlorine was the largest, but if you look at the hexachlorophene, that one didn't do anything. There's no zone of inhibition. If we look at the Pseudomonas, the only one that had a zone is the chlorine. The other 3 were not beneficial in this case. So the effectiveness of chemical antimicrobials against endospores and mycobacteria are very important to look at, because these are the ones that can survive most things. Okay, and so when we look here, a lot of these don't actually help against both. Okay, in the sense of mercury, it's not going to be active or helpful on either of these. The phenols are going to be good against mycobacteria, but not endospores. When you look at chlorines, though, they are fair for both. They're a good potential option. We also see the glutaraldehyde is fair against endospores, but it's good against mycobacteria, so it's a good option as well if that's what you're looking at treating.